Hey 7th graders, today's lesson is 2.3 traits. Your learning target for today, I can explain why organisms have 2 copies of a gene for each trait. So in chapter 1 we really focused a lot on how genes make proteins and why those proteins are important as far as their structure and their function goes. In chapter 2 we've been really exploring kind of how we get those genes and how that passes down to make different proteins. So your learning target for today, I can explain why organisms have 2 copies of a gene for each trait. Okay, in your OneNote today, you should have added key concept number 7 to your key concepts page. An organism has 2 copies of a gene for each feature. And again, today we're going to kind of explore why do we have 2 copies? Where do they come from? Okay. Moving on to your bell ringer, if a strand of DNA has a mutation, what are 3 possible ways in which the protein can change? We dug into this a lot in 2.2. Okay, we looked at building the connects and doing all those different mutations. We actually learned of 4 different things that can happen. Technically, only 3 of them are changed, so as the 4th one is that nothing happens when we mutate that DNA, it stays the same. So what are the 3 possible changes we can see? Okay, so answer that there. If you guys need to, take a second, pause this video, complete your notebook and your bell ringer. And when you're ready, hit play and we'll move on. All right, for activity 3, you need to pause this video and watch this video. Okay, mutations and new traits. So take a second, pause and do that. All right, I hope you guys enjoy that video. Okay, moving on to part 4 of our simulation. You guys are going to be looking at different proteins that make bristles on the legs of our spiders that we've been talking about. So here we have Anne, if you guys remember. She makes only one type of protein for the bristle feature. So if we look at the key, you can kind of see her little protein highlighted right there. It's a little like, I don't even know how, it looks like a ramp to me. Okay, so we got little ramp proteins floating around in there, or the bristle protein number 1. Okay, and you guys can see her genotype right there and right here. So you guys can see here's the gene on each of her chromosomes. Remember, we get 1 chromosome for mom, 1 chromosome for dad. Okay, here is Leo on the other hand, and Leo makes 2 types of protein for the bristle feature. So you guys can see he's got the little ramp, but he's also got the number 2 protein, and you guys can see that his bristles are sparse. So what you guys will be doing with this is this little simulation activity. Your link for the sim is right here, and if you guys recall, it looks like this. Okay, so mission 1, comparing spiders. Use the sim to gather evidence about why some organisms make 1 type of protein for a feature while other organisms make 2. So we're going to kind of dig into, what's different in their genes or their genetics that cause one, like Anne, to make just 1 type of protein, or Leo who makes 2 types of protein. Why is that? Okay, you guys obviously at home are not working with a partner, so you guys are going to be both partner A and B. So choose a feature to focus on. You can select any feature except body size. So when you go in here, body size, which is right, body size is on there, is out of the question. So don't even use body size. Okay. And then you guys will work through partner A and partner B. So partner A, by observing the cells for each spider, find a spider that makes only 1 type of protein. So your first goal is to find 1 spider that just makes 1 type of protein. So if you chose the bristle type, which we already know, again, don't do bristle type. Okay, we know that Anne only makes 1 type. So if I pull up Anne's cells, I can see only the protein number 1. So that's what you guys are going to do. So do something other than bristle as well. Okay. And then once you guys have done that, find another spider that makes 2 types of protein. So again, we know with bristle, Leo makes 2 types of protein. So you would find something like that where he has like 2 different shapes of protein floating around in his cells. Okay. So first of all, you're going to go ahead and say the feature you observe. Again, bristle is kind of off limits because we already know this. Okay. So choose either silk flexibility, silk stickiness, venom, body color, or stripe color. Okay, so highlight the one that you guys chose. So if I chose silk flexibility, then I would go through, I would find a name of a spider that only has the 1 type and highlight it, and then the name of the spider that makes 2 types of proteins for that trait and highlight it. Okay, then you're going to answer this question. In the space below, list ideas why you think your first spider makes only 1 type of protein while the second spider makes 2 types of protein. So really take a look at each of those spiders when you guys are in there, like if I'm looking at bristles. Okay, what's different about Anne's information that's different than Leo's? Okay, so if you guys aren't sure, go in and check those spiders out and look for differences other than the number of proteins or the types of proteins. Okay, then you guys will do mission number 2. Changing the spider's genes. You will play the role of both partners again. So work with your partner to change your spider's genes so that both spiders produce the same protein. So you can either make your one spider that only has one type of protein produce two, or you guys can make your two type of protein spider make only one. So that's your goal there. Okay. What did you do to make the spiders produce the same protein? So you really might need to dig around in the sim if you don't know how to change that. Okay, how did this change the affect the spider's traits? So what did you change, and how did that change affect the spider's traits? Okay, then moving on part 6, you guys are going to do a little modeling. So if you guys need, go ahead, pause, complete your guys' sim on 4 and 5. Okay, and then come back to this video. Okay, on part 6, you're going to model kind of what you guys observed with that and tie it all together. So in chapter 2, you guys have been investigating the relationship between genes and traits. Use the modeling tool, variation in spider offspring model, to show how your current thinking applies to Darwin's bark spider's offspring. Follow the instructions below. So your goal is to show why the spider offspring have different traits for silk flexibility. So we're focusing just in on the trait for silk flexibility and looking at why we have different variations. So your do now is to carefully review the proteins each spider has. Use this information to decide which gene versions they have. If you guys need to, you guys can use the sim for this. Again, you guys will be looking at silk flexibility. So if you guys look at this, you guys would be able to see, all right, with silk flexibility, I need to go review maybe just what proteins are available. So this is protein 1, protein 2, protein 3, because you guys will need to know that. Okay, add annotations to explain why each spider has the gene version you recorded. Don't worry about the annotations. I actually just ask you guys these questions. So try to use the following science words in your annotations: gene versions, heterozygous, homozygous, traits, and proteins. Remember, instead of gene versions, we're going to call them alleles. Okay, all right, so here's what your modeling tool looks like. It's on slide 13. Okay, so we have our 2 chromosomes right here with our 2 little genes for our silk flexibility highlighted, and then pointing to each of those genes, you guys have these circles. Okay, what you guys are going to be doing is filling in what gene version or allele you guys think belong in each circle. So if I look at this first one, I'll do this one for you guys. So offspring A has low silk flexibility. Okay, and if I notice, I have all of protein number 1. Okay, so because I have all of protein number 1, I'm going to assume that I just have F1, F1. So I'm going to drag an F1 and another F1 down here. And then up here, I'm going to type in the genotype. So remember, we get 1 copy of our gene from mom and 1 copy of our gene from dad. And that forms the offspring's genotype. So then I'm going to go up here and I'm going to type the genotype, and my genotype would be F1 from mom, F1 from dad. So that's the genotype. So then you guys would need to look at, all right, here's offspring B. You guys can see it's got medium silk flexibility. It's got type 1 and type 2. This one just has type 1. This one has type 2 and type 3. So you guys are going to figure out what alleles they would have, 1 from each parent, and then you guys would type that individual offspring's genotype up here. Remember, the genotype has 2 parts to it. It has 1 for mom, 1 representing dad. Okay, so make sure you guys do all the genotypes and this, and then instead of those annotations, you guys have these questions right here. Okay, so how did you decide which alleles to place with each spider? And so those are the little circles. How did you guys decide whether F1, F2, or F3 went in each of these circles? Okay, and how did these different gene versions or alleles result in different traits for silk flexibility among the offspring? So if we think about their genes, how do we know which proteins they're going to make, and how does that contribute to their silk flexibility? For instance, an example would be, how did the 2 alleles you designated to offspring 1 result in low silk flexibility? So explain your answer to that there. Okay, finally, do your wrap up. Do you have any questions, yes or no? If you guys have questions, type them below. If not, leave that blank. And then 4, 3, 2, 1, how do you think you did? You are done. Don't forget to hand this in, and I hope you guys have a great day. Bye.